commitments. They all have other commitments. So some of these events will be midday here in the library. Others may be later in the afternoon. And we've even done some events in partnership with the public library and done those in the evening so that we can kind of contact out to our surrounding community. So try to be aware of that. We'll keep pushing out announcements of each of the events as they come along. Our next event is going to be at midday, 12 noon on February the 24th. We've got Dr. Jason DeVito from mathematics. He's promised me that he's going to try to make this something that even I can understand, but he does some very high-end mathematical research, and I'm kind of fascinated to see what he's up to. And then in March, we're going to have Dr. Rasha Tiwari from the Ag Department here as well. So be sure to follow those up as you go along. I want to thank Damon Ballou, who's back there running the video feed. IT Services has been a good partner for us on these series, so we're going to continue to work on the ways that we can get these content out to different places. I do want to encourage you, though, to come to the events if you can. It's all too easy these days to tune into a Facebook feed or a Zoom meeting, and really this is an opportunity not only to learn what we're going to hear today as part of the talk, but also an opportunity to meet with each other and talk about the things that you're interested in, maybe research projects that you're working on, maybe research that you want to suggest that you could partner with someone on. It doesn't matter if you're a faculty member or a student or a staff person. That's really part of what UCMARC is doing. Fomenting those ideas and supporting them and encouraging them really is what this series is also about. So take some time to chat with your neighbors when we're done today. Today's speaker, Dr. Saman.
Call me Dr. Saman. I mean, just Saman.
Just Saman.
Just Saman.
For those that don't know, he has a bachelor's and first master's degree from overseas, and then relocated to Florida, of all places.
Yes, sir.
To get his second master's at the University of Miami, and then got his doctorate at Florida International. He's held postdoc appointments both at the Georgia Institute of Technology, Georgia Tech, and UCLA before coming here to UT Martin in 2020. We were chatting about what it's like for people like the two of us to make the move to a new place during COVID, which is always kind of interesting. Your entire interview online with me. So have a chance to chat with him. He's going to talk about the work that he's been doing with pediatric brain injury, particularly amongst younger people. So this is part of the work that's going on based at our campus, but involving researchers from around the world in what's known as the Brain Autonomy and Resiliency Lab. And so he's going to talk a little bit more about that, but perhaps you can help me welcome Dr. Sargolzaei.
Thank you very much. Thank you. Probably easier if I relocate myself here so I can see the slide also. Thanks everyone for coming here. I'd like to thank Dr. Nordberg and Dr. Cavalier and their team for organizing the event and having me presenting here. So my talk is going to be categorized in 3 portions. And the first portion, I want to start telling you a story. It's called The Story from a Neuron to a Neural Network. And once we fall asleep, after that story, I'll start with the rest of the talk, which is going to be about pediatric brain injury. And at the end, I'll be having a glimpse of student-driven research and development projects that we have in the lab. All right, without further ado, let's start with this story. So once upon a time, there was this single cell called neuron, right? That for so many years, is it too loud? Is it good? Okay. So for so many years, there was this belief that neuron can do anything, and there was just this single fundamental units in the brain, right? Just such a great kingdom. Right? Well, it wasn't until Ramon Cajal, a Spanish scientist, recognized the diversity of different cells that we have in the brain, right? He's called the father of, or the architect of neuron doctrine. He proposed the concept of discrete elements that we have in the brain. And more importantly, as I said, he suggested that we have a diversity of different cell shapes and types. If each one of them, they have their own functionality in the brain. So neurons are not alone, okay? But let me tell you a little bit, just spread rumors about this Ramon Cajal. Probably you know him, but he had many school transfers. Okay? It was very interesting to read about it because of the anti-authoritarian attitude. Even at some point, he destroyed their neighbor fence, ended up in prison time. But later, he became an avid painter, artist, and gymnast, but his father had another plan. So his father wanted him to be an anatomist, right? Which he ended up being a university professor, ended up winning the Nobel Prize in 1906. Okay? But the interesting fact is that he didn't win the Nobel Prize alone. As you can see, it's just half a prize. Okay? The other half a prize was for his opponent, just like we have different ideology, right? There was an opponent to Ramon Cajal, and his name was Camillo Golgi. I'm sure you know about it, most of it, about the Golgi staining technique. That's what he's famous for. Well, Camillo Golgi was an Italian scientist. Nowadays, we have a village that it's named after his, because of his effort, because of his honor. And he had also an interesting life event. He had some financial pressure, ended up with being a hospital chief medical officer. He set up his research lab in the hospital kitchen and ended up with staining techniques that we know him. But to focus our discussion, on one side we have Golgi, on the other side we have this Ramon Cajal. Ramon Cajal was saying that, okay, there are discrete elements in our brain, meaning that we have different cell types, that they're not all connected to each other. Under different dynamics, they connect with each other through synapses. Camillo Golgi had another idea. He was suggesting that there is a continuity in the structure and the function of the brain cells. Okay? We call that Reticular Theory. So that was the other half of the Nobel Prize. So the Nobel Prize in 1906 was shared between these two famous scientists. Okay. One was suggesting that there is a reticular theory. The other one is a neuroimaging theory. In other words, one was suggesting that there are discrete elements that are working together. The other one was suggesting, no, there is a continuum of the cells that are working and functioning together in our brain. Now, over time, the evidences of the neuroimaging was collected and was valued more than the reticular theory. Okay? So the idea of having synapses in the brain and the communication between discrete elements was more come to spotlight with more evidence. Even he, at his Nobel Prize statement, he mentioned that however opposed it may seem to the popular tendency to individualize the elements, I cannot abandon the idea of unitary action of nervous system. Good statement. All right. Nevertheless, coming back to our focus of the discussion, neurons are not alone. There is this network of cells that are functioning together that makes our cognitive decision-making and making other functions that we have. So have they lived happily after that? Well, obviously that wasn't the case, right? Because we have all these sort of diseases, neurological disorders, events that is happening to that network that is making things far more complex. Now, learning about the brain network in the concept of the network or neuroimaging wasn't really a thing until recently because of the advent of neuroimaging techniques, right? So you may have heard about the magnetic resonance imaging, right? Electroencephalography, which is looking at the electrical activity of the brain, and transcranial magnetic stimulation, which is going to be not just observing what is happening in the brain or how the cells are functioning, but also applying some forces and change the way that they interact with each other, which is the idea of transcranial magnetic stimulation. Because of the technology advancements and because of the investments, national investments, you may have heard about the Connectome project, which was which was on the spotlight a few years ago. There are governments that are investing in this idea of mapping the brain network, okay, or how the brain cells are connected or functioning together. And there are many initiatives that are going on. But we're just beginning scratching the surface, and there are many things, many more that we need to invest and learn about. Last year, what I have done was I looked at the scholarly activity when it comes to brain network. Okay? So what you're seeing on the x-axis is the number of years, right? So for example, you don't see any scholarly activity terms brain network, and these are PubMed index articles. Starting from before 1970. Starting 1970, you see a rise of interest in this era of neural network or brain network. Okay? And that was because the invention of, for example, MEG or MRI, the techniques, computer tomography or PET imaging. And at the same time, it was because of the computing technology was more enhanced over time. Okay? Now, you may ask, okay, what is that red and what is the blue? What I've done is to look at the difference between younger population, younger than 19 years old, or older population. And you can see that there is definitely a gap between the number of scholarly activity that is focused on younger versus older adults, okay? Now, the reason, because of the reason of that gap, it comes to natural and technical challenges, okay? The same natural and technical challenges come if you're looking at the difference in the number of studies between male and female, right? You're definitely going to find that gender bias in the scientific studies. Now, things are getting better, but we still have that gap between the older and younger of age. Now, I told you about the natural challenge, and I will tell you about the technical challenge in this. The natural challenge is that the physics of the brain is changing, right? Just like our body grows from birth or before birth, the brain size is also changing. We're having more number of cells or being added to the brain, or more number of cells that are also being eliminated. We'll talk about that addition and elimination factor. But one challenge could be that natural challenge. The other natural challenge is not only the structure of the brain is changing very rapidly during the early ages prior to birth or after birth, but also their functionality has been changing depending on the environment that we're growing up, right? Depending on the stimulus environment that we have. For example, this is a study in 1994. There are a series of those studies that they're looking at the brain structure and function for birds. And they reported that the birds that they store food and then retrieve that location, they have a larger hippocampus. Now, hippocampus in our brain is responsible for memory formation, that sort of thing, right? So you can imagine that the more the birds is inclined towards storing and retrieving memory, they have a larger hippocampus, okay? And also they have more neurons and fewer apoptotic cells. Now, apoptotic cells are the cells that they are responsible for the death, right? We have two pathways in the brain. We have plasticity, we have death pathways. Plasticity is I'm learning something new, and that is plasticity, it's changing. Death, and when I started working in this era, I was surprised at why do we need that death pathway, right? All right, so there is a natural challenge. There is and also not just the macro type of changes that we're seeing in the larger scale, but also in terms of the synapses, these connections that are forming between different cells in the brain, between different neurons in the brain, that is happening in the scale of hours. Some synapses are being added over time, some of them are eliminated. You know, and that there is a peak of addition and elimination of these synapses, these connections, right after birth, okay? Or just before birth. Apart from the natural challenge that we have in studying brain networks in children or in younger population, there's a technical challenge. So that study right on the top right corner, they looked at the number of successful MRI sessions in children. And they reported that, okay, most of the time we're staying at 50 or a little bit above 50% success rate when it comes to imaging children in the MRI. And that makes sense because the MRI is a very noisy environment, and it makes the children to scare and move. And what would happen to the MRI signal is going to be died because of movement. You're going to have trash data if you have movements in the MRI, okay? That's why they tell you to just stay with no movement. So that is a technical challenge. Another source of technical challenge is the softwares that we have, right? Or neuroimaging packages that we have. Most of the time, they're developed with the adult population in mind. And when it comes to analyzing children neuroimaging data, we're going to see some biases. We're going to see errors, and that's a study that I've done in 2015, that not all the softwares are a good fit when it comes to the developmental brain or children's brain. All right. So put it all together, studying neuroimaging in children is like hitting a moving target. If you've ever tried hitting a moving target, but it's not even just a clear path of tracking. It's just all these sort of deviations from the path. All right, are we ready for the next part? Okay, the second part, I'm going to be more specific about brain injury and how preclinical studies, animal studies, can help us to learn a better perspective about what would happen to the brain after there is a traumatic injury. CDC defines traumatic brain injury as a disruption in the normal function of the brain that can be caused by a dump, blow, or jolt to the head. Everyone is at the risk of TBI, especially children and older adults, right? And that makes sense. There are reports of 2.2 million emergency department visits because of the TBI, and leading into 50,000 deaths. But the problem with brain injury is not just that incident of the injury, but it's more because of the consequences of that injury, right? For example, when, in this 5-year outcomes of the study of the outcomes with person with TBI, 22% of them were died, 30% became worse, 22% stayed same, and only 26% improved. So the problem is not just that incident, it's a sequel of an event that is happening after the injury time, right? After injury, people are 50 times more likely to experience seizure. They're 11 times more likely to accidental drug poisoning, and you can see infection and pneumonia as an example of what is happening. But apart from these statistics, the main concern with the traumatic brain injury type of event is that it happens to an individual, and the sequel can be broadcasted to the society, to the family of that individual for a long time. So, take-home message at this point, TBI is a very complex disease with short and long-term sequelae. Prevention is the best treatment, obviously. Now, I told you about all these sequelae, but what can we do to study traumatic brain injury in terms of the science behind it, and how can we intervene to reduce the impact over time, okay? TBI is one of those conditions that is, again, it's very complex because if I have a TBI, it's going to be definitely different from someone else having TBI. Not only because of the type of the injury, but also because I'm coming from a different neurological background than someone else. Everyone has this neurological and genetic background that is unique to themselves. So when the TBI is happening, it adds more complexity, right? But Because of all those issues and because of the ethical issues, most of what we know about TBI comes down to preclinical studies, animal studies. Now here I wanted to share with you something, it's called a new outreach initiative. It's called Come See Our Ward. Okay? If you just Google Come See Our Ward, it's an initiative, outreach initiative, that is telling us about what's going on in the world of animal studies. Okay? And I guess I appreciate what they're doing is to show that not all animal studies are crude or that sort of thing, but I think they're trying to broadcast a real picture of what's going on in the animal studies world. Okay? For example, they're reporting something, some interesting facts. For example, 30 Nobel Prize needed rats for the awarded contribution to science, something like that. And these stats are probably now updated, but what else? A rat can fall as far as 50 feet and land unharmed. Okay? I don't know who tested that. So having animal studies in the era of traumatic brain injury can help us to closely study 3 different perspectives of the injury incident and after that. We can look at the behavioral perspective, what would happen at the molecular level, and what would happen to the system level. Now, I'll share with you an example of all these 3 events or 3 layers of studying brain injury. One of the things that is happening after brain injury is the chance of having memory impairment. Right? Now, to study memory impairment and at the same time to come up with medications that are helping that memory impairment to improve over time, there is a simple test among many tests that can be done in the preclinical world that is called novel object recognition. So rats are very curious animals, right? If you know them, if you put them in a new place, if they're not under stress and they feel safe, they start to turn around, they start to look for new objects, they start to investigate their surrounding, right? So in this novel object recognition, what is happening is that we put the rat into a box with 2 similar objects, right? That is the training aspect. And the rat starts to learn about the shape, learn about the smoothness of that object, okay? Now, since we have 2 similar objects, right, you can imagine that they're going to spend equally amount of time investigating these 2 objects because they're similar, right? So 50% on average in this one, 50% on average on that one. Now, you take the rat to their home cage and leave them for a while, depending on what type of memory you want to study. And then after a while, bring them back into the cage. Now, this time, you're going to put a new object. Okay? Now, obviously, the hypothesis is that since they're remembering the old object, they're going to start investigating and spending more time learning about the new object if their memory is intact. If there is a memory impairment, then they're not going to recognize the new object and kind of just juggle around, right? This study was published by our group at UCLA, and the postdoc before me published this study. What she reported is the animals that they don't experience traumatic brain injury, they're going to spend an average almost 70% of their time here interacting with the new object, because now they remember the old object, they're not remembering the new object. Another question? Oh, sorry. Thank you. But injured animals, it stays at 50%. So 50% on the new, 50% on the old. You don't see that new object impact on this interaction with the new object, right? Now, to tell you about this one, this is a medication that they're using, D-cycloserine. So there is a treatment that we're using to treat the animals with the injury, and that helped that novel object recognition rate coming back to around 65. D-cycloserine. Yeah. Now, we talked about the behavior. Now let's talk about the molecular level. As I said, there are different pathways, like all these proteins, for some of you may be known, but all these proteins, they're responsible for a specific task when it comes to in the downstream microbiology, right, downstream biology. Some of them are responsible for plasticity. Some of them are responsible for the path, for the death. We call them pro-death pathway. Now, after traumatic brain injury, you see more of these proteins activated that are responsible for the death of the cell. So that's why you're seeing these, for example, JNK, nNOS, or P38 being more active as the death signaling pathway. Means that they will lead to the death of the cell. Right? After brain injury, there are reports that, or there are findings that these are more activated. Sorry. These are more activated than the plasticity pathway. Apoptotic cells, yes, yes. Mm-hmm. Now, behavioral, molecular, now let's go to the brain. And, we were at the brain, but now let's talk about the circuit. So what I started studying was to look at the MRI of these brains after injury, right, or before injury, or with or without injury, and seeing what is happening in terms of the functional MRI, in terms of how the brain is connected in the internal circuit, how they're connected to each other, and if we can find a difference between the injured and non-injured. So we've had an MRI session run 4 days after injury and 14 days after injury. Well, it shows the postnatal day, but they were 23 days old, and then at the 14 days after injury, they're 33 days old. And yes, there are MRI machines for animals. And for example, a human, the strength of the magnetic field is about 3 to 3.5 Tesla. For the animals, it's going to much, much higher in terms of the strength, just to make sure that we're catching all those incidents. I don't know if we have a sound, but that sort of MRI magnetic gradients are changing that rapid to just catch all these images that we have in the MRI. And that's the other one. But here's what I found, and these data that I'm sharing is not yet published or peer-reviewed, but I guess they are eye-opening in terms of, on the top, what we're seeing is a group of animals without injury. On the bottom, we're seeing the group of animals that had experienced injury. Okay? The location of the injury model was right here on the top here. Right? And what we're seeing on the control animals' brain, the warmer the color, means that they're connected to each other. Okay? They're communicating with each other. That's a surrogate index of connectivity. If we see this correlation between the signal between different locations, but at the injured animals, we see that lack of connectivity or lack of correlation on the site of the injury when compared to the rest of the brain, which is showing as the loss of connectivity, and it can be studied further to understand what is going on and whether that medication is helping to improve that connectivity over time. Okay? As you can see, from 4 days to 14 days, there are more warm colors that are shaping at the site of the injury, so that is a good sign. Over time, the brain is going to try to recover from the injury. That's at least an example, or at least a hypothesis that can be checked further. Okay? We can also do graph theoretical analysis. Now, the graph theory, if you have heard about it, it's helping to model all these connectivities together and analyze, for example, what is the traffic, how much of the communication we have in that model or in that graph, or because so that's going to be a little bit off the topic, but our brain is is trying its best to accommodate all the requested resources, to accommodate all the requests with limited amount of energy, okay? So they're trying to optimize everything, right? That's the idea of the small word network in the graph jargon of graph theory. But what we can do with the brain network connectivity is to look at how the brain is handling the resources or allocating resources after the injury event. Is it still following that small word network model, which is addressing all the requests with optimized amount of energy or not? All right, so preclinical studies of TBI, they complement clinical studies, providing granular level of understanding of pathology and mechanisms. That's the second take-home message. Now, from this point on, I'll tell you a little bit about some of the projects that is going on. And before that, I want to share this slide, probably one of my top 3 slides today. But I've had the opportunity to work with a group of students since I joined UT Martin 2 years ago. And they're the reason of the success of some of our projects that we're doing. We have Julia that is recently joining us virtually. She's in Poland, but she's having this IEEE scholarship that's doing research with me. Tiffany Decker is a current student in mechatronics. Erin Evans, she's also our student here in electrical engineering. Katie Hunt, student athlete in the UTM women's soccer team. She's working with me also on a few projects. Kaylin Patel, mechatronics, and Abigail Plank, which graduated. She's currently at Vanderbilt, and I'm very proud of her. I'm looking forward to hear more about her accomplishments in Vanderbilt. But most of the projects, development projects that we're doing in the lab, it comes to the word of virtual reality, right? So to tell you a little bit about the virtual reality, it's a computer-generated graphical representation of the real fantasy world. Now, you may have heard different words like virtual reality, augmented reality, and then you're hearing mixed reality or extended reality. Each one of them has their own specific. Like virtual reality is everything is animation. You're not really in the real world. You're all surrounded with graphics, animations, cartoons. Augmented reality is the one that you place these animations onto the real world and then look at the lens, you'll see that animation in the real world. Mixed reality is when you're having, when it's hard to really recognize the difference between that animation and the real world, and they try to exchange information with each other. That's a mixed reality or extended reality. Okay? Now the field of virtual reality has been here for a long time, but recently it comes to the attention because of because of the computing technology is evolving very rapidly, but more importantly because of the strength that it gives us in terms of the control of viewpoint, right? When you're looking when you're in the movie theater, you're looking into the one single dimension, and your perspective is constrained to the perspective of how the director wants you to see that, right? Versus virtual reality or extended reality, you as a user have that capacity to change your perspective and look at things from different perspectives. Here are a few screenshots that I took in our campus, right? So you can bring these digital graphics or animations right to your classroom or to your environment, and that sort of thing that can be done when it comes to the power of virtual reality. Here's an example of one of the projects that we're working together with Tennessee Corn, and thankful for the support that Dean Winters and their team provided us with, is what we're trying to do with the Tennessee Corn through the lens of virtual reality is to broadcast the experience of the corn farming, from planting, from the growth of and from the harvesting season, from different perspectives. So we're working on, Katie and Erin are working on that at the moment. We're working on develop 2 products. One is 360 tools where you can actually just go to the farm at different time points and see what is happening and then gain information. We're also developing a three-dimensional game that you can test your knowledge by following the corn maze. So you just pass the corn maze and answer some questions about the corn and that sort of thing. So that's an example of the use of virtual reality in farming or in agricultural education, let's put it that way. Another one that we're working on is neuroimaging in the field of autonomous vehicle. Now, autonomous vehicles are coming to the roads, right? But if you ask me whether I feel safe to let the car drive me, probably I would say no at the beginning, and that sort of thing. But there are many questions unanswered when it comes to the new technology just bringing it to the roads, right? Without answering those, it may not feel safe. One of them is the autonomous vehicles. Obviously, having an autonomous vehicle and then study or do research on that is expensive. So what we have done is we're looking at games and virtual reality world to study objectively using, for example, neuroimaging techniques, what is happening in the brain when you experience riding an autonomous vehicle. That is the objective of that study. And recently, we started looking into a robotic approach to mask use in the COVID era. And the idea is to let the artificial intelligence, for example, scan through and tell you, okay, for example, this guy doesn't have a mask. Why don't you put your mask on? And that sort of thing. So that is a project with the computer science department. We recently started looking into that. And now the work of the artificial intelligence, obviously this project is starting because of COVID, but it will last after that. For example, there are some receptions that you can train models to act as reception. You can train the model to act as a reception in a department or in a building. That sort of thing can be done to continue this work. That's all I have. So thank you so much for taking a moment of your busy daily life and joining me and learn about us. And I'll be glad to entertain any questions. Yes, in one. Sure. Sure.
So every night I go home and watch TV. I see all these ads and all these supplements, brain supplements. What are your thoughts? You chose one with the
Cycle serum, yeah.
What are your thoughts?
Yeah, well, good question. So Dean Winters is asking about, seeing all these ads about brain supplements and what do I think about it. I can tell you my own experience. I used to take multivitamins, lots of them, because my mom asked me to do so. And for a long time, I didn't know if they're effective or not. Now, you may know that some of these, and by no means I'm trying to give medical advice, but there are FDA routines for that is not probably happening for most of the supplements, right? And they have their own certifications and that sort of thing. Now, when it comes to brain supplements, I know that, at least I've heard about things that exist that helps to memory information, and to some extent they're valid, but I think, at least in my opinion, is that I am in favor of natural things, such as the idea of brain plasticity, right? Making your brain think or work with your brain cells by practicing things, thinking about things, analyzing things, studying, that sort of thing is a better supplement to help the brain function. Just like because we have a term called experience-dependent plasticity of the brain, which means that our brain learns from experiences. And the more we have productive experiences, obviously our brain is going to function better. And prevent from neurological disorders.
So you would say that me learning a new language would be better than me taking L theory?
Yes, I think it's more effective. I think it's more effective. That's a good point.
Where's your research going to lead from here? What do you plan to do?
Yeah, so good question. So we're planting the seeds, and every day, it comes just like virtual reality. Every day comes a new application domain that we're thinking of. Probably Dr. Neep is already tired of me just coming up to his office and talking about the new idea I'm thinking of. But to answer your question, I guess we're just starting. And lots of these things happen because of the financial supports, like startup grants our college has, or Tennessee Corn provides us with that financial support department, and national foundations to just, so as far as there is that financial support, and as far as we're able to recruit new team members, new students, we're able to cultivate the culture of research does not have to be something really fancy and changing the world all of a sudden. One of the advice that I got from one of my supervisors was that he said in the first day of my postdoc was, Saman, we're changing things in a millimeter scale. So don't think that you're changing things rapidly, and as long as we're in the right direction. So as long as we're able to recruit more students and financial supports, I guess we're going to just keep continuing what we're doing right now. Yes. Yeah, so if I understand it, so the question was, what are my thoughts on applying these studies into autistic brain and autism, the world of autism? That is a very good domain. And obviously, autism is one of those, just like traumatic brain injury and how complexity is, autism is exactly like that in terms of complexity. And they're doing that sort of brain connectivity network and neuroimaging studies. I know a group at UCLA that they're working on that, and they're sharing the data. And one of the advantages of living in 2022 now is that you have access to these multi-centers data. They're trying to be open source in terms of their data, obviously anonymized, but they're and different data. They're sharing the data, and we can work on that. And that's definitely a valid application and a beneficial application to look at the autism data. And there are interesting facts when it comes to brain connectivity network in autism versus controls, that they have a very formalized way of allocating resources in the brain when it comes to autistic children. And you see that in the behavior, obviously. So yes, definitely that is a good domain.
Are there any good models for autism?
For autism, I think there are different things. I guess it's interesting when it comes, interesting and upsetting that when it comes to neurological disorders, what we're trying to do is we're always behind the disease most of the time. We're trying to see the impact of a disease after it happened. And I think we're at that stage at the moment for most of these conditions. But there are good amount of data for autism.
Great. Well, I hope you'll hang around and talk with Dr. Sargolzaei. There's also cookies and refreshments over here, but I hope that you'll share your applause again for a very good
Thank you very much. Appreciate it. Thank you. Appreciate it.